Hello, I think it's recording now. I would like to present my poster in about 5 minutes, so I will go straight into that. My name is Fernando Fierro. I'm an associate professor at UC Davis, and the title of this work is "A Cell Culture Model Suggests a Key Function for Paracrine Signaling in the Development of Bone Fibrous Dysplasia." So very quickly, I know how to move slides. Fibrous dysplasia of the bone is characterized by hyperactivity of this subunit of the GPCR receptors called GS alpha, when the mutations are gain of function mutations that lead to accumulation of cyclic AMP due to this hyperactivity of this GS alpha. These are mostly found due to two specific mutations. Either arginine 201 is replaced by a C or an H. So to model the disease in a dish, what we did was to overexpress using human bone marrow-derived mesenchymal stem cells, stromal cells, either the wild-type version of this GS alpha that is coded in a gene called GNAS, or the mutants. And as you can see here in this figure 1, they actually show that characteristic accumulation of cyclic AMP, while the overexpression of the normal GS alpha doesn't lead to a significant increase, probably because the other subunits are still limiting how much cyclic AMP can be generated. And this is just to show that this gene is actually regulated during osteogenic differentiation. So when MSCs are cultured in osteogenic media and will become osteoblasts precipitating calcium, you can see that there's almost a 3-fold increase during the first 2 weeks of this gene, suggesting that it probably plays an important role during bone formation. This is a characteristic morphology we see in the cells. The MSCs, as you know, have typically fibroblastic morphology, and that wasn't affected by expression of the wild-type GS alpha. However, when you express the mutant version, these cells become, have this very different shape with a very distinct stress fibers. These are actin bundles, characteristics typically of an excess of Rho A signaling, which has been already described to be a downstream of this excess of cyclic AMP. And that, of course, leads to impaired migration. Those cells have very poor cell motility. We performed video microscopy for that. You see that control cells or expressing wild-type GS alpha have normal cell motility, which is strongly downregulated in the cells expressing the mutant GS alpha. As we were suggesting at the beginning, there's an important role for this protein in bone formation. However, the expression of this mutant actually causes a strong downregulation of bone formation, measured as alkaline phosphatase on day 10. This is an early marker of basically pre-osteoblast. And then alizarin red, which stains the calcium deposition, the mineralization measured at day 21. And as you can see here, there is a strong downregulation of bone formation in these mutants, which could well correlate with the observation that patients having this condition have a more fragile bone. Interestingly, when we look at proliferation, the cells expressing mutant GS alpha show actually a downregulation. There's also some effect caused by the wild type overexpression of GS alpha. However, when we grow these cells in co-culture with MSCs that are not genetically modified, because this, I forgot to mention, this condition is typically post-somatic, the mutations acquired. So people express often both, some percent of cells normal GS alpha and others with this mutation. And you can see that when they are grown in co-culture, these two types of cells and normal cells with the cells that have been genetically modified, there is actually no differences in cell proliferation. And even more, if you take now the supernatant of the mutant cells and give that to normal cells, you see that the supernatant of the mutant cells is causing a significant increase in proliferation, strongly suggesting that there is some paracrine effect exerted by the cells expressing mutant GS-alpha that leads to increased proliferation of normal cells. Yeah. So in this assay specifically, there are no, what is measured is not the cells that are genetically modified. These are normal cells that just were exposed to the conditioned media, the supernatant, produced by cells that have this modifications. This is very quickly just to show you that actually we know that this is a wind reporter system to see canonical wind. Basically, the more GFP, it means there was more binding of beta-catenin to their target sites in the nucleus. And you can see that the control or overexpression of wild type doesn't cause much of a, there's almost a doubling of this canonical wind signaling observed in the cells exposed, again, just to the supernatant of these mutant cells. And to look at what is in these mutant cells, we run preliminary proteomic analysis. This still needs to be confirmed by Western blot analysis, but some of the key proteins are found here. You see, for example, VEGF, an important angiogenic factor, is expressed at a certain level in control and cells expressing wild type, but that is over more than a twofold increased in cells expressing the mutant GS-alpha. So I hope you enjoyed this poster, and thank you very much for your attention.